Hello, everyone. We're glad to participate in RPPW 21. We thank the scientific committee to offer the opportunity to present our work on point rhythms in active reception by humans and other animals. By active reception or probing, information about the environment is provided through the effect of objects on a self-generated signal or probe. The procedure can take various forms. The most well-known is the man-made radar using electromagnetic energy. Another one is the sonar that mimics echolocation by bats. But bats are not the only animals to use sonar. So do some marine mammals such as dolphins and sperm whales. But my favorite model is that of electrolocation by weakly electric fish, such as the elephant nosefish. The animal possesses a specialized organ that generates the so-called electric organ discharge, or EOD. The feedback of the current flowing through nearby objects gives information about their composition, their location, orientation, and helps them to find their prey, just as bats find insects and dolphins fish. The bats and cetaceans' ultrasonic transients are called creaks. The elephant fish's electric transients are called pulse discharges. If such pulses or transients are considered all or none, they form sequences of pulse intervals, which I call point rhythms. Point rhythms are defined from the sole time series of successive inner pulse interval durations. Many different systems generate such one-dimensional signals, as for instance, human finger tapping, neuron firing, or even cars passing at all. Today, inspiration will come from the model of sperm whales' clicking behavior. Sperm whales are huge mammals who dive really deep in the ocean to feed on squids and other fish in an environment with no light to see anything. Sperm whales emit time series of ultrasonic clicks, which have been separated into two types, the regular clicks and the creaks. The regular clicks, they're separated by intervals lasting around 1 second. Here are the regular clicks with echoes. The creaks, they're much faster and more variable series of weaker clicks, separated by intervals lasting around 100 milliseconds. Here's some creaking with regular clicks in the background. Because there's no way to use a camera to observe sperm whales when they dive hundreds and hundreds of meters deep, a team of cetologists equipped them with a whole set of sensors to record at which depth they swim, their moves, their changes of orientation, tilting, etc., and also their click production. They observed that the creaks are produced mainly in a preferred layer of the sea, supposed to be their hunting zone, where live the squids. They use regular clicks when they are close to the surface and when they navigate between the surface and the depth. The authors propose that the creaks are used for a finer discrimination of the prey position before catching them. Indeed, similar acceleration has been observed in bats reaching insect prey and also in dolphins when they attain a target, the final buzz. Elephant fish Elodie also accelerate in rate when the fish navigates closer to its foraging spot. So we consider that creaks are a mean to get a finer spatial resolution through a higher sampling rate for better resolution when searching and reaching a target, while slower regular clicks are enough while hanging on at the surface or for diving navigation. We hypothesize that any adaptive agent would similarly accelerate a point rhythm of probing when searching and discriminating by comparison with mere waiting or navigating in open space. So we asked blindfolded human participants to navigate, to search, and to discriminate targets over a large table by listening to the sound feedback of their finger tapping. The sound depended on layers of paper covering the table. A focus area mimicked the hunting zone. The hunting area contained disc targets. In addition, each disc covered a thicker diameter core positioned either vertically or horizontally, placed underneath the disc target. The fingertip was inserted in a thimble that enhanced sound production. The participant first waits 1 minute for a change that triggers the navigation phase. After the hunting zone has been reached, she searches for the discs. Once a disc is detected, she must discriminate the right position of its diameter core, and finally search for the two other targets and also discriminate the core positions. Here is the tapping profile for one participant, along time on the x-axis. The y-axis gives the inter-tap intervals. The higher the rate, the shorter the interval durations. You see that the rate remains low, longer intervals on the left at the time of waiting and navigating in the wide open space. And you see the highest rates, shortest intervals at the time of discriminating within the discs, with intermediate level while searching the targets within the brown hunting layer. General results are very significant and confirm the individual profile example. Again, the inner tap intervals on the y-axis, shortest intervals at the final discrimination phase, longer intervals when waiting and navigating, acceleration when searching in the brown hunting zone, and even more when discriminating. In conclusion, the point rhythm model demonstrates similar probing behaviors in humans and sperm whales, together with bats, dolphins, and electric fish, using higher rates when finer discrimination becomes crucial. We hope that the heuristics of the etho-mimetic approach will offer stimulating avenues for rhythm prediction and perception studies.